So I guess we can start. At first, hello to everyone, to our participants. This is the third webinar in our Synergy webinar series about animal welfare. It's a collaborative effort of all three cost actions: LIFT, Improve, and AFFECT-EVO. All these cost actions have animal welfare as their central theme. Today, it is my great pleasure to introduce Associate Professor Laura Webb from the Animal Production Systems Group, Wageningen University and Research, where she investigates the sustainability of animal production. Laura plays a key leadership role in several European initiatives on animal welfare. She is a vice chair of the Cost Action LIFT, wherein she is a co-leader of Working Group 2 and two sub-working groups that focuses on the time scales and effect balance. She is also actively involved in the EU partnership on animal health welfare and leads the action on positive animal welfare, which works to validate indicators of positive emotions in pigs, cattle, and poultry. Her research focuses on positive emotional states and the accumulation of affective experiences over time in vertebrates as well as on sentience in invertebrates, such as insects, bivalves, and cephalopods, like squids and cuttlefish. A line of work she describes as both fascinating and fun for her. Drawing inspiration from positive psychology, her work contributes significantly to our understanding of what makes animals just not survive but thrive. Please join me in warmly welcome Laura Webb, who will be speaking today on the animal happiness, understanding and assessment through behavior, cognition, and physiology. So Laura, the floor is yours. You may start.
Thank you so much, Roesh. Thank you for the introduction, and thank you very much for inviting me to give this webinar. I didn't know it was the third one, so it's very exciting. Before I start, I would like to sort of start with acknowledgements, because this is obviously not my work. I haven't been doing this work all by myself. There's many people involved, and I'll have a slide at the end with all the names of everyone. But just to start off by saying, this is the work of many very enthusiastic and smart individuals that deserve credit. So please don't think this is all my own work. And in addition to that, I'd just like to mention that when I talk about happiness in this presentation, I mean a long-term state. So maybe you could conceptualize it as how one feels most of the time. So it's not a short-term state, like joy, for example, which might be considered more like a short-term emotion. So basically, the work that we've been doing is aimed at opening a window into the emotional minds of animals, for example, into the mind of this caterpillar here. So here you see the brain of a caterpillar with the stained dopamine neurons. And this is really what is the essence of what we do. So we try to understand and then also assess affective states, mental states in lots of animals. And when we look at invertebrates, another question is, do these animals have the capacity to feel in the first place? And to do this, we use three fields of research, so behavior, cognition, and neurophysiology. And you see here some examples. On the right side, you have a bee brain, and, some octopamine neurons stained, which is the equivalent of adrenaline in insects. And you have in the middle there a cricket performing a cognitive test, in this case, a motivational trade-off test. And on the left side, you have a camera filming cow-calf contact. So these are examples of behavior, cognition, and neurophysiology. So just an outline of what I don't know if you can see this box in the top hand corner of my screen, hiding the outline. I don't know if you see this, but I'll just...
No.
Oh, you don't see, it's just me then. Okay, great. So just an outline so you know what I'm going to cover today. So I'm going to focus first on some theory about the concept of time and how that relates or might relate to affective states, drawing a lot of inspiration from human research. And that has to do with some of the work that we've been doing at the LIFT. And then in connection to that, dive into the concept of happiness in humans and whether this might be just a human construct or maybe it has a biological underpinning, which might potentially suggest that we could find it in other species. And then I'll look at some examples of how to assess the short-term event-based emotions in behavior, cognition, and neurophysiology with a small note about rigorous research and validity. And then finally, just touch upon some potential tools to assess something longer than emotion, something like cumulative affect or happiness in animals. And we will look at 3 domains, and that's the neurophysiological biomarkers, affect balance, both of which are covered by the LIFT, with different subgroups of the LIFT, and finally, outcomes of happiness. So I hope that there's something new in this for everyone, and I hope that you enjoy this webinar. And let's get started. So I'll first start looking at the concept of time. Just give you a small reminder here. Oh, first I wanted to say that I will not cover in the outline, I don't cover how to promote positive states in animals or how happiness is exactly assessed in humans. That's like a whole thing in itself. Right, so time. So just a quick recap, just in case some of you don't or are not familiar with this. This is the way that I will use the terms emotion, mood, and affect. So emotions are short-lived object or event-based experiences that are either pleasant or unpleasant and vary in arousal. And the running mean of emotion is sometimes called mood. So mood is like less intense, more long-term, and I'll just show you some graphs after this that illustrate this. And finally, affect is often used as the umbrella term for both emotions and moods. And when it comes to affective states, we are very much biased towards negatives, not only in our research, but also in our perception. So a lot of the research in animal welfare is focused on negatives, and only recently have we switched our attention to also including the promotion, understanding, and assessment of positive states, but also in our own perception, negatives weigh more, and this is important to remember, and I'll come back to that towards the end of this webinar. So you can see this illustrated in these images here at the bottom. Now, trying to put a timestamp on different affective states is very difficult. And this is an example of what was suggested in humans. Obviously, there's a lot of individual difference in this and also context difference, but it has been suggested that the emotions in humans last from seconds to days, while moods might last from hours to weeks, maybe touching upon months. And then the question is here, where do we put something like happiness, something that is even longer term than mood? And the suggestion is that this will be around here. Happiness is suggested to be more stable than mood, but less stable than personality. So it would be somewhere in the middle here. A way to consider and think about the concept of time when we talk about affective states is to create such graphs. So this is a graph created by Mike Mendel and colleagues, and you can see that they've put emotion on this black line. So you see these peaks are basically the emotional events, and these are linked to the prediction that someone makes of outcomes and whether these predictions are met or above expectations or below expectations. And then the blue line that I've added here, because the gray line was not very easy to see here on these slides, this would be mood. So you can see it's less intense, it's less variable, this could be considered more long-term, and it's not based on events, but it's basically the running mean of emotion. Another way of illustrating this would be in this way. So this is something that we created back in 2019 in a paper entitled What is Animal Happiness? And here you see again these lines of emotion that vary more quickly in time, although you see also variation here. And then mood is a bit slower and less variable. And then you have this third line here, which is referred to as affective happiness. The idea, and these are the 3 affective states that, to my knowledge, have been described in humans. but not in animals, as far as I know, or not as much in animals. And this affective happiness line here is in fact considered to be stable in stable context. So it's, as you can see here, much more stable than mood. But again, and I'll explain this later, also less stable than personality. So it does vary. But this is a way of representing these things in time. And of course, many, many questions remain. Just have to move this camera again. So when it comes to emotions versus moods, is there evidence in animals that they experience these as distinct processes? Can we, as researchers, distinguish between the two, or, is that difficult to do? Does it matter for welfare whether they're experiencing emotions and moods? Can we just assess emotions, or do we also have to assess moods? And again, what is the timing? Because we know we have some idea of the timing in humans, although again, as I said, it's very difficult to, assess. But what about other species, right? So this might be very different. I think they showed relatively recently that flies have a different perception of time than we do. And when it comes to a longer state of integration, like what I currently call affective happiness, is this just a human construct? Is there a biological underpinning? And if humans do have biological happiness, then which other species might have it too? And also, why might it have evolved in the first place? So what might be the function of having this third state? So I'd like to now dive into the concept of affective happiness and also other forms of happiness in human. I should say at this stage, so I started by saying that I will define happiness as how one feels most of the time. This is the definition of affective happiness. But in human or positive psychology, there are many, many definitions connected to the term happiness. There's also something called cognitive happiness, which has to do with how a person evaluates how their life is compared to how they expected their life to be. So you can imagine this requires quite high cognitive abilities. And there's another form of happiness, I call them all happiness, but there's different words in the human literature, which has to do with whether you think your life has purpose or meaning. And I basically tried to collate as much evidence as possible that there might be a biological underpinning to affective happiness or other forms of happiness in humans. But this is quite difficult to do. As you can imagine, a lot of the human research is correlational. So it's trying to show relationships between things, but we don't know the causal, the direction of the causal link. So as I said, happiness has been found, and this obviously depends on how it's assessed. So I said I wouldn't go into that. So we're just going to assume that the way that it is assessed is always is reliable. But happiness is both relatively stable and somewhat variable. So it's less variable than mood, but it's more stable than personality. But personality accounts for a big part of happiness. This depends again on the context and the study and the method and so on. So that's why there's a huge variation here in the impact of personality on happiness. But it has been shown that if you look at what is sometimes called in studies long-term happiness, or apparently there was also different timescales there, the personality effect increases. And there's a lot of studies looking into the potential heritability, so the genetic basis of happiness, and there's been evidence, there seems to be evidence that's a genetic basis for it. And the idea, and these are quite old theories, but a lot of theories were suggesting that you have a kind of genetic base point. You have a baseline around which you fluctuate, but you come back to this through adaptation following, important life events that did affect your happiness level. So you tend to go back to your default state. Another piece of potential evidence for biological underpinning is that throughout countries and across the globe and in all societies, there seems to be a U-shape relationship between age and happiness. So you see here in this graph, age is depicted at the bottom in years, so from 16 years old to 68 years old. And then on the y-axis, you have the happiness score. And here you have 3 lines, which represent 3 different types of happiness. So the red one, I think, is affective happiness. The blue one is cognitive happiness. And the green one is meaning of life. But I'd have to check exactly how they assess these things. And they did this in 145 countries, I think, in another study. But the idea is that there's a U shape. So you are happier in your childhood and in your later years. And then there's a dip in the middle, which some refer to this as midlife crisis. And, you know, this seems to be universal. So it's very interesting. And there is even a paper that looked at this effect in, I can't remember which grade ape, I think it was chimpanzees. And they also studied happiness in these captive chimpanzees, and they seem to have found the same U-shaped relationship. But you'd have to go and read that paper and see how they assess happiness. In these great apes, I think that's super interesting also. I think one of the questions to the keepers was how happy would you be if you were this individual? So that's an interesting assessment method. And then a little bit more evidence. So a lot of people suggest there's something called a positivity offset, so that everyone is a little bit happy across countries, no matter what the situation of the country is, suggesting, you know, less impact of environmental factors versus your own personality and genetics. So people are always a little bit mildly happy, we could say. And this could suggest that there's an evolutionary function to this, and this has evolved for a particular purpose. And what these purposes might be, it's very difficult to establish, but some suggest that it increases your propensity to be social, gives you stronger social relationships, makes you more likely to reproduce and spread your genes, and it is also seems to be predictive of future health and longevity. And then there's neuroscience evidence that happier people, certain brain areas are more plastic. And this seems to be specifically true for reward areas of the brain. So all in all, this is all a bit difficult to disentangle, and it's not very strong evidence, but I think there's a potential suggestion to a biological underpinning for the human affective happiness. And the question now is, okay, but can we find this in animals? Is there evidence that it exists in animals? Does it matter whether it exists in animals? So all these things we need to study further and think about. But now I'm going to end a little bit the part on happiness and move to emotion. So these short-term event-based states, and try to look at, instead of the understanding, move towards the assessment. So how can we assess animal emotions? And I'm going to focus on the positive, I think, mostly. And yeah, I just picked out a few examples. Obviously, I can't cover everything, but there's a lot of work being done on this, I think, throughout Europe and beyond, and also the European partnership that Roesch mentioned at the start is also doing a lot of work on this in cattle, poultry, and pigs. But first, a little bit about looking at affective states across species. So we have humans, and humans can verbally, a lot of humans can verbally report self-report how they feel. And when we move away from humans to more distant species, this becomes more difficult. And so how do we approach this? Well, we do a lot of rigorous research, right? We design careful tests and criteria, and we try to establish whether these beings also might have similar emotion-like states. And of course, we're all very careful not to be too anthropomorphic in our research, right? And what happens if you read, it's very interesting, and I really advise everyone to do this. Read papers on emotion through different species. So if you work, for example, on rodents, go and read the human literature and go read the invertebrate literature. It's very interesting. You'll see how different it is. So when we have humans, you have a child laughing, you'll very easily see in literature just words like laughter, and this means joy, and there won't be a lot of explanation as to how they came to this conclusion. And this makes sense in a way because we're studying our own species, right? So it does make sense to be a little bit quicker in our conclusions maybe, but yeah, you'll see the way that they do the research and come to conclusions is very different to what we do, for example, in farmed mammals. In the mammal, you might also observe a certain vocalization. And what do? You do a lot of rigorous testing, and then you'll be careful with your conclusions and the words that you use. And you'll see the vocabulary is different. So they won't say laughter, they'll say laughter like. And then they'll say it's mostly linked to positive emotion, but not in all contexts, or we're not completely sure. And you move away and you get to even more distant species like insects. It's not even about affective states anymore. It's about demonstrating that they have the capacity to feel. So are they sentient? And a lot of this research focuses on pain. And again, they'll use different terminology. The conclusions will be more and more careful. And then you could question whether we should also go and study the plant literature. And there's this really wonderful book about plant cognition, behavior, cognition, and intelligence. And I really advise you to read this book. It made me think a lot about animals and the way we define things. And then certain definitions, for example, for behavior can be applicable to plants, or definitions of cognition could be applicable to plants. So it's very interesting to go and read this book and realize what we're really looking at and how much plants can do, in fact. Okay, so coming back to how we assess emotion, you probably know that emotion has, well, let's say 4 parts to it. So it's linked to specific changes. And there's a part that we can't access, and that's the subjective component. But we have 3 fields that we can use to try to, assess emotion, and that's behavior, cognition, and neurophysiology. And I've just listed here a few examples of what you might study. For example, to look at emotion in the neurophysiological component, you might look at neuroendocrine markers, cardiovascular markers, immune markers, metabolic markers, but you might also look at a combination of these, what we call composite biomarkers that combine lots of different biomarkers into one score, like the allostatic load, for example. So in behavior, you can also look at different things like body parts or whole body behavior, but also things like behavioral diversity that are also a sort of composite score, or even things like group dynamics. And I'm sure a lot of you are familiar with proposed cognitive indicators of animal emotion. And these include the famous judgment bias, the attention bias. Memory bias is a bit less studied, but there might also be other tests. I think also sensitivity to reward loss is one of them, for example. But I will come back to that in the next slides. So here you can see, so I wanted to focus a little bit on the positive side of things. And these are examples of vocalizations in various farm animals that have been recorded in what would be assumed to be positive context. So you can see the grayish boxes are the spectrogram, and the graph on top of that is the waveform. And if you look at I think it was the pig, the goat, and the chicken, these 3 calls are expressed in a situation of food reward, while the cattle, horse, and sheep calls shown here have to do with reunion with a young one or a peer. And you can see sort of similar patterns to some of these. And that was very interesting to see that across these species, and it includes a bird, but it's mostly mammals, there are similarities in the way that they express themselves in potentially positive contexts. And we found that positive vocalizations are often shorter with a lower, less variable fundamental frequency. And vocalizations, I think, deserve a lot of work in terms of assessing both positive and negative emotions in those more vocal species, especially because some species are a bit less vocal. And then there's the famous play behavior with its 5 criteria. So play is often used as an indicator of joy, emotion like joy. And play includes 3 different types of play. At least 3 different types of play have been described in the animal world, and that's social play, locomotory play, and object play. And to validate that a specific behavior is play, we as researchers have come up, not me, but others have come up with 5 criteria. It should be not functional. It should differ in form from a functional behavior, but be similar. It should be repeated in time, but not stereotypical. It should be voluntary, spontaneous, but also self-rewarding. And it should be initiated in relaxed and not stressed states. And I want to show you examples of play behavior, not in maybe the species we are more familiar with, but in insects. And in fact, all 3 of these play categories have been described in an insect. Social play in a form of play fighting was described in wasps, social wasps. Locomotory play, I'm going to show you in the next slide, and objects play in the next one. So this is potential play behavior observed in the fruit fly. It's a super funny video. And basically here you have this red circle, and this is a carousel. So it's a platform that's just spinning. And then you have a green circle there, and that's a food source, food reward. And the video is accelerated. The yellow part is the tracking of the fly flying around the arena. But just look at what it does. So it spends quite a bit of time on this carousel. It does also feed, of course. Sometimes it has to think a little bit about what it's going to do next. I think it goes again, yeah. So it's an interesting behavior, right? There's no function to it, or the no function that we can come up with, yet they seem to be doing it repetitively in these kinds of tests. So this is a potentially, if we would follow those 5 criteria that I mentioned, we would potentially come with the conclusion that this could be described as play behavior. Oh, and another study in bumblebees this time looked at object play, and you can see here what kind of testing arena they had. So you can see on the middle part of the slide the nest, and then they had a tunnel with a path leading to a sugar reward. So they could just walk straight through the testing arena and go to the sugar reward and then go back. On the way, though, you can see that there's a field with balls, wooden balls, of varying colors. And what happens is that every single bee will roll these balls around, and they won't do it once. They'll do it over and over and over again. The frequency with which they do this varies between individuals, and it seems that the males do it more than the females, and that there's also an age effect that the younger bees do it more, which is also consistent with what we observe in mammals and in humans. Okay, so I talked a little bit about behavioral indicators of positive emotions, and now I'd like to switch to cognition. But I'd like to state at this point that we're not, I would say we're not really sure what these cognitive tests are assessing, sorry. So is it emotion, or is it mood, or something else? For example, what we observe in the judgment bias literature is that I think the way that they are testing the animals and the way that they are changing the animals' lives, the assumption is that it's about something long-term, right? There's a long-term enrichment process going on, and then there's a testing on the judgment bias. But if you look at judgment bias testing in insects, the treatment is applied just before the testing. So is that then short term, or is it, so I think we need to think about what these tests are exactly assessing and whether it matters or not what they are assessing. This video shows an example of another test called or it's a test that assesses sensitivity to reward loss. So I don't know if you're familiar with this test, but basically animals, they get a big reward and then the reward size decreases, they'll be less motivated to do the test. And vice versa, right? If the reward increases, they're more motivated or faster to do the test. And this is affected by their living conditions. So it's thought to be potentially able to especially detect states like, well, let's say negative welfare. So it's maybe more on the negative side. But this is a pig doing this test in a runway. It's just a funny video. It doesn't really add much to the presentation. But it's a very highly motivated pig. Very happy to get this reward. And this is what's work done by my colleagues here at Wageningen University. And I'd like to show you oh yeah, and so this is some of the work that we did in the European Partnership. So this is my student, Tamara Kedves, and she went through the human literature looking for possible links between positive emotion and cognition to see whether we could gain inspiration for further cognitive indicators beyond the ones that we've already described in the animal literature. And she came up with all of these. I could show you the detailed results of this because we're just about to submit the manuscript. But yeah, as soon as it's published, I hope that you will be able to read it. And I think I was very inspired with some of the stuff that she found. And here you can see some examples of the different things that she found. And of course, because we were looking at humans, we also had things like language, but other things can be more applicable to animals. Okay, this is not really about emotion, but I just thought it was really cool to show you too. And this is just cognition in bumblebees, and it's really fun. It's basically showing how bees can learn tasks from each other. So here in this video, you see the bee with the red dot is the bee that has learned the task. This is the experienced bee. And it's learned that if it pulls on this string, that it gets access to this blue disc. And in the center of this blue disc, there is a sugar water, so a reward. And this other bee next to it is an inexperienced bee learning this behavior from this bee. And they learn really fast. And what you see in the graph next to the video is how this will go, this will cross through generations. So they will really learn this, and they basically develop what you could call a kind of culture, and they teach each other this skill and this task. So bees are really fun animals to work with. And this is another cool example that could be evidence that bees are aware of their own body size, which is also really cool. So you can see here in these two videos a bee, and then in the middle of the arena, a wall with an opening in it. And you see in the video on the left, the opening is much larger than on the video on the right. And you'll see what this bee does. So in the first video, the opening is big enough, and the bee will sort of assess the situation and go through relatively quickly. But in the second video, it's going to do something different. And you can see here, it's just gonna turn to the side and then go through. How cool is that? Okay, so moving on, because I see I've only got 10 minutes left, or maybe a bit more. Neurophysiology. I actually, in the outline, I said I would focus on behavior and cognition, I think, or somewhere I said that, but I will also say a little bit about neurophysiology. So as I mentioned, you can look at different areas of neurophysiology, like neuroendocrine, cardiovascular, etc. And there's been some suggestion as to some promising indicators of either emotion, or I would say maybe even cumulative affect. And you can look at single versus composite markers. What I want to say here is that for animals that do not do very much in terms of behavior, it is very difficult to come up with behavioral indicators or cognitive indicators. For example, with bivalves, which, I mean, their behavior is to open and close their shell, right? So it's very difficult to study them. with these two fields, but of course you can always look at physiology. They have a heart, they have, they breathe. And so what we're doing now, and this is work done by my colleague at Wageningen Marine Research. He's developed a sensor that he can put on the shells of these bivalves, and he can record heart rate. And from that, we can deduce heart rate variability. And we're going to look at whether we can see heart rate variability being also an indicator of something like stress in these animals, the way that we see it in mammals. And this is some of the work done in insects. So I think these are mostly bee brains, and you can see some of the neurons that have been stained here. Dopamine, which is linked potentially to positive experiences, and oxypamine, which is linked to excitation and arousal. And also these animals have incredible capacity for learning and memory. So this is also really cool research. And I also have a colleague here at Wageningen University who is a neurobiologist in caterpillars and butterflies mostly, which is also very interesting. And just to close the, let's say, more short-term assessment part, short-term affective state assessment part, we shouldn't forget that we have to obviously do high quality research. We have to be rigorous in what we do. We're not working with subjects that are able to report how they feel. We don't have a gold standard. So we need to be careful. We need to, at all costs, avoid circular reasoning. And we really need to follow these validity procedures, construct, external, internal validity. I also show you here some of the tests you can do. These tests were proposed by Mike Mendel and Georgia Mason. I think they're writing a book, or they've maybe published already a book on this. Very important, careful validation steps for affective states in animals. And I think it's also important to say, and I think it's great that we're in this webinar, right? It is like a synergy of 3 cost actions, because I think what we really need to do is to share knowledge, share skills, share facilities, come together, minimize experiments, but really also make the most of them, not reinvent the wheel, and really do high quality research together. And that's what we're doing in these, in the LIFT and also in the European Partnership. We address some of these steps of construct validity in identifying novel, not per se novel, but in validating, let's say, indicators of positive emotion in farm animals. Okay, so moving on to the third and last part of this talk, I would like to look more at the long-term states. So potentially we could call it happiness, or just long-term accumulation and balance of positive and negative states, or just cumulative affect. So just a quick reminder that we don't know whether animals have a state of affective happiness. We think they probably have some sort of cumulative affect, so that goes beyond emotion. So I'm just going to talk about cumulative affect here. And I'm not going to distinguish between mood and anything longer than that. And looking at humans for inspiration, this is also work that we do at the LIFT. Looking at humans for inspiration about this, we identified 3 different categories of methodologies that we could draw from to assess, let's say, long-term accumulation of affect in other animals. And the first one is affect balance. And this involves essentially the counting of short-term emotions over time, and whereby the researchers have to compute this integration themselves. Then you have indicators of cumulative affect from neurophysiology, whereby the body of the animal is computing the integration, and then you're just recording it. And then there's something called outcomes of happiness, which are actually correlates of cumulative affect. So it's not the same level of assessment. So I don't know if you saw this during the LIFT workshop already in Utrecht, but basically we have a target here, that's the rabbit, right? So we want to look at cumulative affect, and we have to look through all these steps, the why, the how, and what can we do, what is feasible to get to our indicator at the end. And now I'm just going to talk about the theory, but basically this should not be forgotten. So we're going to stay at the why level. But again, we have these two estimates of cumulative affect. So this affect balance and the neurophysiology, and I'm going to describe them. Again, this is not my own work. This is the work of many people combined. And this is actually the work of a sub-working group in which I'm not involved. So I got this slide from someone else, but these are potential neurophysiological markers of cumulative affect in animals that were derived from human research mostly, but also some animal work. And one of them is brain plasticity. For example, hippocampal volume has been suggested as affected by cumulative affect. Epigenetics is another one. Gut microbiota, but this is a very, very complex, difficult one. Even in humans, we don't fully understand what's going on there. Then you have immunology and oxidative stress, with some examples pointing there, but there will be a full paper about this. And finally, telomere attrition, that I think a lot of you are already familiar with, is another example here of a marker of cumulative affect, whereby the body is doing the accumulation. And I'll show you how it can be, like what it requires in practical terms. But first, I'll tell you what affect balance is. So this is the other one. So affect balance is basically the affective experience of the animal, which has to do with the accumulation and balance of the positive and the negative affect over time. And what we did is we went to the human literature and we tried to look at through all these methods and try to extract conclusions, let's say, from these methods. And what we can conclude is that almost in every single study on affect, I would say even every study in affect balance, they look at both positive and negative affect, right? They're not just focused on the positive. They look at both, and they look at both separately first. And then they integrate both. And the way they integrate these is different per study. So it could be a ratio, positive over negative, or it could be a sum, positive minus negative. It also depends on the participants of the study and the aim of the study. Sometimes they want to focus on depression, like negative states, and they'll do another calculation. It seems that they're trying to derive proportion of time spent in particular valence states. So proportion of time feeling positive or proportion of time feeling negative. And they don't so much focus on things like duration or intensity, because humans are able to report simply, how often they felt positive in the past week. It's based on both looking at the present moment, so how do you feel right now, but also on recall, like how did you feel in the past 2 weeks? Sometimes I ask someone, how do you feel right now 7 times per day for a week, and then compute that. And then they sometimes ask 7 times a day or just at one time to report how they feel. So there's all kinds of different methods. And then when we take this and apply it to animals, there's so many questions. For example, does the type of affect matter? In humans, it's not taken into account. They don't care if it's joy or contentment. They put it all into the positive side. Does duration and intensity matter when we look at animals? Because it's different. How do we integrate things? What matters? What kind of time should we look at? Minimum time period, and what is the minimal sampling frequency, and so on. So many questions. But I just wanted to also show you this slide, which is a little bit debatable, but it's also super fun. So if you look at humans, what you notice is that happy individuals report 3 times more positives than negatives. So there's a 3-to-1 ratio. And for happy marriages, this is even 5-to-1, right? That's how much the negative weighs in human life. If you have a 2-to-1 ratio, you're doing okay. You're getting by. If you have a 1-to-1, you are in a depression. You are clinically depressed. So when we say that a life, I think it was a neutral life in animals is equal positives and negatives, this might be completely wrong. This might be a very, very bad life. And when we say a life worth living is a little bit more positives, maybe not. Maybe that's the neutral one, So I think we need to really think about these things. And this is just to show you how you would assess affect balance versus cumulative neurophysiology. So, sorry, the circles here would be like all the counting of all the emotions, whereas the stars would be like these 2 sampling points. So you'd have to sample less often, and the body would be doing the integration work, but this would probably be potentially be an intrusive measurement, whereas with affect balance, you could just record, for example, vocalizations in a non-intrusive, non-invasive manner. And just to finish very quickly on the third category that I mentioned, which is more like correlates of cumulative affect. This is again from humans, and this is based on the broaden and build theory. So this is Barbara Fredrickson's work, and she sort of states, and there's empirical research to support this, by the way, that positive emotions build up over time, and they lead to a broadening of one's perspective, which makes them more open and curious towards the world and leads them to build what she calls personal resources. But this is basically strong personal relationships, what else? Resilience, also physiological resilience, mental resilience, building competences, skills, and so on. So, and that will lead this like a sort of upward spiral. So you go through this and over again towards the state of high well-being or flourishing. So just to summarize, there is so much we don't know. And I think we're doing a great job trying to figure things out, but there's still a lot of work to be done. And we need to think about, just not only the assessment, but also really understanding what is going on. What are we trying to assess? Does it matter? And how are we going to then assess it? And do we want intrusive methods or not? And so on. So that's a bit of the summary of the situation here. And do we want to assess short-term states or long-term states? Do we care whether animals have effective happiness and so on? So many questions remain to be answered. And as I said, I think we need to work together and share knowledge and skills and really make the most of what we can do. I think I will end with my acknowledgement slide. So I just want to say thank you to so many people. You can see here the three sub-working groups of the LIFT, from which most of this work was extracted, but also my colleague at the entomology group, Alex Haverkamp. Some of the slides were his and the videos and so on insects. And then my student, Tamara, but also the people at the European Partnership for Animal Health and Welfare, whom I did not put here. But so many people to thank. Thank you so much for your attention.
Thank you, Laura. Thank you so much.